All right, BIM 100 students, welcome. With finals coming up fast, we put this together to help you absolutely nail the core concepts from Professor Pasquinelli's first 3 lectures. Think of this as your high-yield study guide. We're going to cover exactly what you need to know.
So let's kick things off with this great quote from the man himself, Francis Crick. It's just a perfect way to remember that this whole field is driven by pure curiosity. It really sets the stage for our dive into the molecules that make life happen.
We have to start at square one, right? And that means the absolute foundation of genetics, the structure of DNA, the blueprint itself. I promise you, if you get this down, everything else that follows is going to make so much more sense. And everything, I mean everything in DNA starts with this little guy, the nucleotide. For the exam, you absolutely have to know its 3 parts by heart. You've got the phosphate group, the deoxyribose sugar, and of course, one of those 4 nitrogenous bases.
Okay, now for those bases. There's a really simple way to classify them. You've got the purines, that's adenine and guanine, and you can see they're the bigger ones with a 2-ring structure. Then you have the pyrimidines, thymine and cytosine, which are a little smaller with just a single ring. It's kind of counterintuitive, but just remember, the shorter word, purine, is the bigger molecule.
So how do we build this famous double helix? Well, it's really just 3 key steps. First, you link all those nucleotides together with super strong phosphodiester bonds. That creates the sugar-phosphate backbone. Then, the 2 strands line up, but here's the trick. They run in opposite directions. We call that anti-parallel. And finally, the bases pair up in a super specific way. A always with T, and G always with C. And just like that, you have the rungs of the ladder.
Okay, so we've got the structure. Now let's talk about how that specific structure gives DNA one of its most critical properties, its stability. And we can actually measure this using something called the melting temperature, or TM for short. It's just the temperature where half the DNA double helix has unwound into single strands.
And this brings us to a classic exam question. Seriously, expect to see something like this. It's all about connecting the structure to its properties. Why does having more G's and C's in a DNA molecule make it more stable and harder to melt?
Well, here's the entire answer on one slide. Look closely. A guanine-cytosine pair, you can see it right there, is held together by 3 hydrogen bonds. Count them. 1, 2, 3.
Now compare that to an adenine-thymine pair. It's only held together by 2 hydrogen bonds. It's really just simple chemistry at this point. It takes more energy, more heat, to break 3 bonds than it does to break 2. That's it. That's why more GC content equals a higher melting temperature.
Okay, so let's apply that. Say you're in the lab and you find 2 DNA samples. One melts at 80 degrees, the other at 90. Could they be from the same organism? The answer is a hard no. No way. That huge difference in melting temp means they must have a really different GC content, and that means they have fundamentally different genetic blueprints.
All right, get ready for a mind-blowing fact. Every single one of your cells has about 2 meters of DNA packed inside it. 2 meters! How in the world does the cell solve this massive packaging problem and cram all of that into a nucleus you can't even see?
Well, the cell's solution is both simple and brilliant. It's called the nucleosome. This is the fundamental unit of DNA packaging. For the exam, the easiest way to think about it is like this. DNA is the thread, and it gets wound around a protein spool made of 8 little proteins called histones.
And the cell doesn't just stop there. It takes these millions of nucleosomes, which look kind of like beads on a string, and then coils them up and folds them and compacts them over and over again until you get this incredibly dense, organized structure we call chromatin.
So that leads to the next big question, right? If the blueprint is packed away that tightly, how does the cell get in there to read a specific gene when it needs to? This is where we get into the super cool topic of gene regulation. It's all about controlling access.
And this slide right here, this is a hugely important comparison. You've got to know this. The cell uses these little chemical tags on the histone proteins to either open or close the DNA. When it adds acetyl groups, that's acetylation, it tends to loosen up the chromatin, making it open so genes can be turned on. On the other hand, adding methyl groups, methylation, usually does the opposite. It compacts the chromatin, making it closed and turning genes off.
Okay, so that all sounds pretty good in theory. But this is science, right? We have to be able to prove it. How can we actually look inside a cell and see if a gene is in an open or closed state? Turns out, there's a really clever experiment that lets us do exactly that.
It's called the DNase protection assay, and the logic behind it is actually pretty simple.
You take an enzyme called DNase that just loves to chew up DNA. Now, if a gene is in an open, accessible state, the enzyme can get right in there and destroy it. But if the gene is packed up tightly in closed chromatin, it's physically protected by those histone proteins, and so its DNA survives. Simple as that.
Okay, let's do a quick rapid-fire recap of the absolute key takeaways from these first 3 lectures. This is the stuff you want to make sure is locked in your brain for the final.
So if you only remember a few things, make it these. The double helix is made of 2 anti-parallel strands. A pairs with T using 2 hydrogen bonds. G pairs with C using 3. Because of those 3 bonds, higher GC content means higher stability, or a higher TM. DNA is packed into chromatin using those nucleosome spools. And finally, for regulation, astility means open or active, methylation means closed or inactive, and the DNase assay is how we test for that.
And I'll leave you with one last thing to think about as you study. We've talked all about the static blueprint, but what about the process of copying it? How does the cell make a perfect copy of all 2 meters of DNA every single time it divides? Well, that's where things get even more amazing. Good luck with your studying, you're going to do great.